Today we're going to talk about meiosis, which is the reduction and division of genetic recombination. We had discussed mitosis the other day on how a cell divides and gets into two identical cells. Well, this is going to be two genetically different cells. Meiosis is described as the form of cell division by which gametes with half the number of chromosomes are produced, and gametes refer to your sex cells. You start with a diploid cell, which contains 2 times, and you cut it in half, which is haploid. Meiosis is sexual reproduction. 2 divisions, meiosis 1 and meiosis 2. And there at the bottom you do see the sperm plus the egg, 23, is haploid cells, produces the zygote, which is a diploid cell. Sex cells divide to produce gametes, sperm or egg. Gametes have half the number of chromosomes. Occurs only in your gonads, testes, or ovaries, and in the male it's called spermatogenesis, and in the female it's oogenesis, which means egg or ova. Spermatogenesis, you see it starts with a 46 chromosome cell, 2N. It splits into 2, and that should be in meiosis 1. You should still have 46 cells, 46 chromosomes in each one of those cells. Then they divide into 23, which contains a haploid. And then the egg, you'll see there that it only produces 1 egg, so it's not like the sperm cell, but you produce just 1 true egg and 3 polar bodies, which will later die. Interphase 1, similar to that of mitosis. Chromosomes replicate in the S phase. Each duplicated chromosome consists of 2 identical sister chromatids attached at their centromeres. Central is the pairs are replicate. In phase 1, the nucleus and the nucleus are visible. There you see a picture. Cell division that reduces the chromosome number by 1/2. 4 phases. You got prophase 1, metaphase 1, anaphase, and telophase 1. Prophase 1 is the longest and most complex phase. 90% of the time there in the cell cycle, chromosomes condense, a synapse occurs, homologous chromosomes coming together form a tetrad. Tetrad is 2 chromosomes of 4 chromatids, sister and non-sister chromatids. Homologous contain DNA that code for the same trait but different versions of those genes. Genes occurs at the same loci or at the same spot on the chromosome. Prophase 1, synapse, you have the sister chromatids, you have the tetrad, and then the sister chromatids on the outside. Pairs of homologous chromosomes are pairs of chromosomes, maternal and paternal, that are similar in shape and size. They carry genes controlling the same inherited traits. Each loci position of gene is in the same position on the homologs. Humans have 23 pairs of homologous chromosomes. First 22 are autosomes, meaning body chromosomes, and the last pair are sex chromosomes. This is your X or your Y. And there you see eye color is located at the same spot along with hair color. Now, in a lot of studies you'll see that hair color and eye color are also multi genes, meaning that there's more than one gene that codes for a specific eye color. Crossing over may occur between non-sister chromatids at sites called the gismata. The crossing over segments of non-sister chromatids break and reattach to other chromatids. The chiasma are where chromosomes touch each other and exchange genes. Now this is called genetic recombination, which this is where most people, this is where they get their differences. These crossing over allows for a lot of genetic variations. Genetic recombinations, you can see that they cross over, so you can see there the variation would be, and this happens in this phase. There you see paternal and maternal, they're crossing over to give you different types of gene combinations. In the female, you are a XX chromosome, and in the male, you're an XY. Meiosis 1, homologous separate. The nucleus disappears, spindle forms just like in mitosis. Metaphase, shortest phase, this is when they all line up in the middle and they'll eventually separate. Homologs line up at the equator or metaphase plate. So you can see the different combinations that you could get right there. In terms of independent assortment, how many different combinations of sperm could a human male produce? 8 million different combinations. And then you multiply that by what the woman could actually give, so you have 8 million times 8 million. So lots of different combinations. At anaphase, they separate and move toward the poles. Here you see the separation occurring. Each pole now has a haploid set of chromosomes. Cytokinesis occurs and 2 haploid daughter cells are formed. Then you have cytokinesis, which is the division of the cytoplasm and all the other organelles. Meiosis 2. No interphase 2 are very short. No DNA replication. Meiosis 2 is similar to mitosis. Same as prophase and mitosis. Nucleus and nucleolus disappear. Chromosomes condense, spindle forms. Same as metaphase. They line up in the middle and they will eventually separate during anaphase, like you see here. And telophase 2 is same as telophase. Nucleolar reform. Cytokinesis occurs, which is the division of the cytoplasm and all the cells organelles. 4 haploid daughter cells are produced, and they are not identical in any means. They're called gametes, which are eggs or sperm. Telophase 2, there you'll see the 4 bodies that's actually being produced. And genetic recombination, remember that happens in the first meiosis part. Important to populations, raw material for natural selection. All organisms are not alike. Strongest, most fit survive to reproduce and pass on their traits. You know, you don't have a slow gazelle passing on traits. What are the 3 sources of genetic recombination or variation? Crossing over, independent assortment, and random fertilization. Cell containing 20 chromosomes at the beginning, meiosis, at its completion, produce cells containing how many chromosomes? That would be 10. It contains half the number. This is a karyotype. It just shows the arrangement of your chromosomes, and if, you know, they can do a karyotype on you to see if there's something wrong with any of your chromosomes. There's a disease called trisomy 21 where there'll be 3 chromosomes show up on the 21st set of chromosomes, or they'll show missing segments of chromosomes. And there's just a full picture of it a little closer, and that's of a female. And there's the Down syndrome trisomy 21 that I was talking about. Instead of having 2 during non-disjunction, they didn't separate, and both from one of the parents, they gave both of those chromosomes instead of just one side. Fusion of sperm and egg is formed what's called a zygote. A zygote is a fertilized egg. Cell containing 40 chromatids at the beginning of meiosis would, at its completion, produce cells containing how many chromosomes? 10.